Can you hear me?
Good afternoon, sir.
Good afternoon. Okay. So, as I told you that our next topic will be biomolecules. Huh? This biomolecules. So, biomolecules means these are all carbon compounds. Okay, all carbon compounds found in the body of organism. Okay, and that is found in the body of the organism, and that is in the living tissue. Right, this tissue is to find out that what are the different compounds, what are the chemicals that are present in the body of the organism. These things are uh, identified in two different methods. One is that for the organic compounds, and another one is for inorganic compound. Say for organic compound, the living tissue is crushed in the acid. That acid is TCA, that is known as trichloroacetic acid. Then the tissue that the compounds, some of the compound, they get dissolved in this trichloroacetic acid, and but then it is that slurry, whatever we get, that is filtered. When it is filtered, then we get that some of the component, they remain on the top, and the other come at the bottom after, that is in the filtrate. Okay, and this one is retenant, that what is not passing through the filter. So this is there is here actually this portion which come out, which pass through this membrane, this filter, that is known as acid soluble. So there is two things we get: acid soluble portion and acid insoluble portion. All right, acid insoluble portion. This portion is for the organic compound. So naturally, what will happen in the acid soluble portion, those which are small molecules, they will come out. Okay, they will come out. These are that simply the biomolecule. They are simply biomolecules, or we can say them because they are micro molecule, and those which remain at the top on the filter, which does not pass through the filter, that is acid insoluble portion. This is the acid insoluble, and this acid insoluble portion is that those which are very large molecule, and they are mainly the polymers, and they have molecular weight. Molecular weight is usually greater than 10,000 Dalton. Okay, greater than 10,000 Dalton. These are very large molecules, and here it is less than, that is less than 1,000. Particularly here, of course, it is in between that 18 to 800 Dalton. This portion, this soluble portion. This way, it is the organic compounds. They are investigated, isolated, and tested. Their percentage, etc., whatever we have to do. The another method is that the mm, analysis of S S analysis. So S analysis is, it is this is used for the inorganic component, particularly the elemental analysis. Here the tissue is first, this tissue tissue is dried. When it is dried, that there will be water will evaporate, is it? It evaporate. So this will be dry, and after that it is burnt. When it is burnt, then all the carbon compound evaporate, mostly in the form of carbon dioxide and water vapor. And the remnant one is the S, and that S is analyzed for the presence of different element and their percentage, etc., that which one is more, which one is less. Okay, so this is the general method that by which we can analyze the mm, the elemental component or the chemical analysis of the tissue of living organism. Now here it is one question, say from here, that biomolecule. So one question is, after doing the chemical analysis of organic compound in living organism, two fractions were observed, namely the two fractions when this chemical analysis, particularly for the organic compound is done. Then two fraction, what are those fraction? Acid soluble pool and acid insoluble pool. Then carbon pool and hydrogen pool, inorganic pool, organic pool, aqueous pool and non-aqueous pool. Which one will be the answer? Is Answer which one? Yes?
Option A, sir.
Yeah. So just now I have explained. So there is no problem. There is one is acid soluble portion and another one is acid insoluble portion. Okay, for the organic compound. And the acid, which acid is used for this analysis? Remember, can you tell? The TCA, trichloroacetic acid. TCA, this is the general method. Similar, another question with that, the other portion. They identify the term as in the in terms of living tissue sample analysis from the statement given below. So what explain this as for the elemental analysis? Okay, these are four options. Four options. Which one is the correct definition or explain the term as? Okay, organic compound oxidize to gaseous form, carbon dioxide and water vapor after burning the tissue. I think this is the correct, is it? Hello, can you hear me?
Yes,
Yes,
sir.
sir.
So this one is correct. Please respond when asking. Otherwise, I feel that you are not listening or attending. Okay. This material left after burning the tissue which contain inorganic element, calcium, magnesium. Okay, all these are, this is true. After burning, these things remain. Okay, the compounds moves in the form of gases. That is not the explaining the S. So whatever is left, that one is S. And compound removed in the form of gases, yeah, this is also take part compound which may be soluble in intracellular fluid. No. So this number A, this is the correct definition for the S. Okay, for inorganic material. Similar, another question is, say here, this one I will say we will discuss first. This after grinding a living tissue in trichloroacetic acid and then staining it, you would obtain two fractions, acid soluble pool and acid insoluble fraction. Acid insoluble fraction does not contain nucleic acid, polysaccharides, lipid, flavonoid, and alkaloids. Which one?
E,
Yes? Which one?
E,
E, A, or please, you read it, which option you are telling? Not clear.
option D.
Option D. Okay. Why it is option D? Say, this is what happened. There is, as I told, that in the filtrate, will be the portion which are small molecules. Okay, and the particularly the polymers, so they are on the in the acid insoluble portion. So acid insoluble portion, actually they are nucleic acid, polysaccharides, then it is protein and lipids. Okay, these four type of molecules, they are found in the acid insoluble portion. This four, and this one, flavonoid and alkaloids, they are the secondary metabolites, and they have molecular weight is very less. Their molecular weight less than 800 Dalton. So small molecules, and these small molecules will come to the in the acid soluble portion. So acid insoluble portion will not contain this flavonoid and alkaloids. This is here the similar question. Compound found in the acid soluble pool have molecular weight ranging from, in the acid soluble pool, molecular weight ranging from which one? A 18 to 800 Dalton, 100 to 800 Dalton, more than 800 Dalton, and none of the above. Which one? It is 18 to those which are in the acid soluble fraction, their molecular weight range from 18 Dalton to 800 Dalton. It is somehow, it is less than, that is 1000. Okay, all. Because in the acid insoluble portion, those which are found, their molecular weight is greater than 10,000. So naturally, all these biomolecules having molecular weight less than 1000 Dalton are, less than 1000 Daltons are macromolecule, micromolecule, biomacromolecule, both A and B. They are biomolecules having molecular weight less than 1000 Daltons are, they are micromolecule. They are simply said when we say biomolecule, biomolecule or micromolecule or biomicromolecule molecules. This one, macromolecules and biomacromolecules, this is synonymous. This one is giving more specific that it is biomacromolecule. This is the better term, that is biomacromolecules. Biomacromolecules, so those in the greater than 10,000. Okay, one question is here. Just now we have learned those that I told. The nucleic acid, polysaccharides, protein and lipids are found in the insoluble portion. One question is that about the lipid. Lipids are found in acid insoluble fraction during the analysis of chemical composition of tissues. Okay. Lipids found in insoluble fraction. Give the reason. Why it is? It has very high molecular weight. Is it? See, lipids are not very high molecular weight. Lipid is not a polymer. Lipid is not a polymer. And molecular weight also, molecular weight is less than 800. Okay, but still this one is lipid molecules are coming with the insoluble fraction where the molecular weight is very high. Along with the other polymers they are found. So what is the cause? It has very high molecular weight. It is a polymer. It has low molecular weight. On grinding, biomolecules are broken into pieces and form insoluble vesicles. Which one do you think correct?
Option D, sir.
Yeah, option D it is. Um, Option D. Why? Say this is it is a polymer. No, this one is not a polymer. Very high molecular weight. No, it has a low molecular weight. It has a low molecular weight. That is true. But the main cause why it is found in the acid insoluble portion, this is due to its nature of the molecule. So, do you remember that when I explained the structure of the plasma membrane, at that time I told you that the plasma membrane is made of lipid molecules. Made up, there is lipid bilayer. Is it? Lipid bilayer is the plasma membrane. And lipid molecules, they have a polar head. This is polar head portion, and this polar head portion is hydrophilic, okay, and there is a non-polar tail, polar tail part, and this tail part is hydrophobic. So hydrophobic portion always try to move away from water. So this portion is that in the plasma membrane to protect this tail part. No, that this plasma membrane, in the plasma membrane, the lipid molecule has to arrange themselves in double layer. By arranging themselves in double layer and keeping the tail on the inner side, they are protecting it from water. All right. So what happened when this, for this organic compound analysis, when this is crashed with the acid, then this plasma membrane or any other membrane within the cell, that membrane broken into pieces. All right. So there is a double layer. This double layer, when it will be broken, it will break into small pieces, and it may come in the form of single layer. But as it is a single layer, they cannot stay in this way, open condition. They have to protect this tail portion will move away, And to stay away from the water, these lipid molecules lipid molecules change into a sphere-like structure. It change into a ball-like structure, and in the ball, in such a way that the all the head part, it is exposed towards the periphery, and all the tails facing inward. At the center of the make themselves at the center of the all the tails, at the center of this hollow sphere, and all the heads, they are exposing on the periphery. Okay, so this way, so actually what happened, these molecules, the lipid molecule does not stay in a single molecule, that a large portion, and they make a small vesicles. So these vesicles cannot pass through the that filter, and it is behaving like the large molecules, and so this one remain with the polymers, other polymers. So this is forms the insoluble vesicle, broken into pieces and form insoluble vesicles. And this is why that the lipid molecule, though their molecular weight is very less in compared to those polymers, they are found along with the polymers in the acid insoluble portion. Okay, next question come to now. Different molecules, those which are found in the acid soluble portion, the organic compounds. There is some molecules are uh, amino acids, okay, different those monosaccharides and oligosaccharides. These molecules, then different nucleoside. These are the organic compound. These are micromolecules. They are found in the acid soluble portions. So now we have to see one by one some of the properties. Okay. Let us one question is amino acids. This question, the amino acids are organic compound and are called alpha amino acids. Okay, They are called alpha amino acids. Why? The
Option
option
B.
option B. Amino
Yes, sir.
acids are organic compounds containing an amino group and an acidic group as substituent on the same carbon. Yes, it is on the same carbon. Option B. This carbon that on which to which this different groups are attached. Okay, that carbon is known as alpha carbon, and this amino acid is the alpha amino acid. Next question on the structure of the amino acid. Okay, here is amino acids are substituent of methane. Name the four substituent group occupying the four valency position. You know methane,
Option.
CH4, is it? In which to the carbon four hydrogen atoms are attached. And there is amino acids substituent methane. No, that is from the methane. You can say it is a derivative of methane. From the methane, it has been synthesized or formed by replacing these hydrogens. Okay, this which one? It will be answer
One. One, sir.
one. Hydrogen carboxyl group, amino group, and a variable group R. The general structure of methane is the general structure of methane is like this. There is one hydrogen in one bond is attached. One is say OOH carboxyl group, and one side it is NH2 amino group, and this one is the this side R group. Okay, and R group can be anything. Okay, this may be simple hydrogen, or it may be any other things. So this is the carboxyl group and the amino group are there. So this one is variable. This is variable. In any amino acid, these three are fixed. Okay, these three are fixed, and this one R is variable. And due to this, due to change of this R group, there is a variety of amino acids. Otherwise, in the amino acid, these three are same. So this one will change and amino acid will change. So this is the answer, that hydrogen carboxyl group, amino group, and a variable group, that is R. And the other options given, two carboxyl groups, one amino group. It may have two carboxyl groups or two amino group, but then it will be in the form of R group. So here the same question what I have told just now. The variety of amino acids are formed on the basis of position of
H.
hydroxyl group. position of carboxyl group, position of hydrogen, position nature of R group. This is that due to variety of amino acid is due to the nature of the R group. So if the R group will change, then there will be different amino acid. This R group can be anything. One has been given here. Say here, name the amino acid A to C correctly. A to C. Portion you can have to make it small. Now whole thing is visible. Okay. So here, this one, this
Glycine.
A has a methyl group. Okay. B has only one hydrogen, and C has the hydroxymethyl group. This hydroxymethyl group. This one in the this is the R. This here it is changing. So what will be the sequence? That is the very simple one is this is the very simple one, that here you can see the variable group is hydrogen, is it? Variable group is hydrogen. In case of this one is glycine. Okay, when it is a methyl group, this one is alanine, and this is the serine. So our option will be A, B, C. A will be, this is A alanine, B glycine, and C serine. So this way it change. Okay. So by changing this R group, we are getting the different types of different amino acids. Here is one, okay, another. One characteristic thing is, as I told, that in the R group, when this R group changes, no? Say this one, that here come to this structure. This is this COOH. This is carboxylic group. This is acidic in nature, is it? Acidic. This one is basic. So if say like this one, this amino acid having here one acidic group, one basic group, and if this one R, say like when it was H, okay, or it is not acidic or basic, then what type of amino acid it will be? This is acidic, this is basic. then this will be a neutral one, is it? And if here in the R group, say some another COOH is there, carboxylic group is there, then acidic portion increase. So this will be a amino acid will be acidic amino acid. And if this basic nature here R is replaced by another amino group or some other basic one, then that will be the basic amino acid. So basic amino acids are neutral is that I told initially, that one. Just now we have seen that here it is this one. This one is replaced by hydrogen. So glycine, it will be neutral, and here if it is change, then valine is, there is a neutral, there is acidic, and there is basic one. Say acidic like glutamic acid. You have to remember some name. These are nothing to do with a lot of explanation is not there. The glutamic acid, the lysine, this is basic, valine, neutral. Okay. Now, one very important characteristic feature of this uh, amino acid is that this this group, no, this carboxylic group and the amino group. Okay, both of these can undergo ionization. All right, means it can be ionized. And there is one character, identify the sweeter ionic form. Sweeter ionic form. Sweeter ion, this is pronunciation little bit sweet, sweeter, sweeter, but this is sweeter ion, sweeter ion. Here, here it is sweeter, it is 2. Okay, this is equivalent to 2. Okay, meaning is 2. So 2 ion or double ionic form. This at a time, if in a molecule at a time, both the ions can be formed. Both the ions means ion, usually we see that ions, when it is ion, they may be positive ion or they may be negative ion, either negative or positive, is it? So, but here that in amino acid is such a molecule in which both the ion can be formed in the same molecule. Both ion, positive ion as well as in the same molecule we can get positive ion as well as negative ion, and this is due to that the property that it both side at the same time can undergo ionization. Okay, it can, the amino group also can undergo ionization, and carboxylic group also can undergo ionization. So This is called sweater ionic compound. Right. So identify the sweater form in the given reversible reaction. This reaction is reversible, and you know this protein and amino acid in the if the pH of the solution or the medium is changed, and in the change medium they so their properties also varies and they behave in different way. So this is due to that they can at a time in a particular pH one or the other undergoes ionization or both. Okay, and that's why which one is the sweater ionic? This is this one B. See here it is only the amino group has undergone ionization, positive ion, but this one is normal neutral. This here it is amino group is a normal, carboxyl group has a negative ion, it has changed. But in this B form, in this B form, that amino group also it has formed a positive ion, and this one has formed a negative ion. So both the ions are present in the B. So what is a B is our answer is choose the correct option. Which one? B is the correct. So here it is in the option C is showing that B. Okay, this so these are the things. Then these molecules will go to one thing with the amino acids, that these amino acids can undergo polymerization. Amino acids polymerize. Okay, polymerize, and this polymer of this amino acid, polymer of amino acid is called what? Do you know what it called? Hmm? It is called polypeptide, not polyamino acid. It is called polypeptide. Okay, polypeptide. So why poly means many? That polymer of amino acid is polypeptide, and polypeptide forms the protein. Okay, so that is amino acid. We can say amino acids make protein. Say there are many amino acids because the R group can change a lot. Uh, and there are variety of amino acids, but for making protein only 20 different amino acids make protein. Okay, only 20 different make protein. This is called polypeptide. Why? In the polypeptide or in protein, amino acids are joined to each other by a bond which is known as peptide bond. Okay, the bond is, so in the protein, the bond between the amino acid is the peptide bond, and the peptide bond is formed with the carboxylic group of one amino acid. So this is carboxylic group, another one, say if two amino acids are to join, here this one is the first amino acid and this is the second amino acid, then the bond is formed between the carboxylic group of the first amino acid and the amino group of the second amino acid. Amino group and carboxylic group. Here, during this formation of the bond, okay, one molecule of water come out. One molecule of water, that from here, okay, one hydrogen, one oxygen, one hydrogen, and one hydrogen will come out, and then a bond will be formed. So here it will remain as a here it will remain, say this one will come out, and from here one hydrogen will remain, and this will form a bond, CO and NH, and water has come out. Then this bond that will be formed, that bond is CONH. This is the peptide bond. Okay, like if again this one will join to the third amino acid, say actually we can write in this way also, NH2 CH R group, say OOH. Okay, then from here again one will come out, one molecule of water, and the COHN will remain. So from here one molecule of water. So by dehydration, okay, that for every bond to form, there is one molecule of water is removed. So in the, you have to mainly remember that this peptide bond is formed between which group? and this during that the water come out, and what is the name of the bond. And one thing you have to always, whenever there is a polypeptide is formed, say during this always there is a fixed thing, so that the bond is formed between carboxyl and amino group. So what happen in the polypeptide chain, in one side there is amino group, and on the other side, other end of the chain, there is a carboxyl group. So this one is known as, on the left we can see it is on the left, this is known as N terminal. N terminal, and this one on the right is a C terminal. Right. So every polypeptide chain has a N terminal and C terminal. Let's see what is, and what happened. This polypeptide, in the polypeptides, hundreds of amino acids are there. Okay, this is that the sequence of the sequence of amino acids. Say this is one amino acid, this is another amino acid, this may be third one, may be the same amino acid, or maybe different amino acid. So in a polypeptide, there is a particular sequence of amino acid. Sequence of amino acids. And this structure of the protein, that is called the primary structure of protein. Next is that this primary structure, this polypeptide, then it change its shape by making bond and folding. Here you can see, this is one question, that what kind of a structure of proteins are shown in this figure given. In this figure, several types of structure has been shown. One is this one, that A, that is a straight line. The one here, that two helical structures are there, means two polypeptides are there. Okay, and here it is that folding in such a way that it is showing the 3D structure, three-dimensional structure, and this one you can see a coiled structure. So this is a straight line one is the primary structure. Different labels are there. Here is at one end, this is the N terminal, and this side is C terminal. Okay, that is what here in this line has been shown, N and C. So this N terminal and C terminal, this is the primary, or we can say first level of protein organization. you can say first level of organization, or you can say first degree of, okay, first degree of organization. Then in the next step, what happened that in this form, in the primary structure, the proteins are not functional. All right. This long chain of amino acid, they fold. They fold. When they fold, they coil like a fold in a spiral. And during this, different types of several types of bonds are formed. So the by this, the uh, length of the protein is decreased. Naturally, length of the polypeptide is decreased, and this spiral is the second degree of organization. Then at this stage also usually the proteins are non-functional. Then there is a further folding, and that is known as third level of organization. In the third level of organization, this molecule, they undergo folding in such a way that they form a 3D structure, three-dimensional structure, and this is that a like a hollow sphere. It forms like a hollow sphere. In these, there is maybe some portion, the secondary level is there in some portion. So this is 3D structure. During this, protein molecules are functional at this third level. At the third level in the 3D condition. Okay. This is, but what happened? This is also not the final stage. Of course, for the sum of the protein, this is the final level of organization. But what we find in most of the proteins, there are more than one polypeptide chain. Most proteins have more than one polypeptide chain. And when more than one polypeptide chains comes together, say this one is 3D structure, another also that 3D structure is coming here, another one is coming here, okay. All these are together forming one functional unit, okay. More than one polypeptide come together in their third level of organization. Each of the polypeptide in their 3D structure, third level of organization. And when that happen, that is the fourth degree or fourth level of organization of organization. So that is called quaternary structure. So primary, secondary, tertiary, and quaternary structure or fourth level of organization. Okay, so here it is given in this question that what kind of a structure of proteins are shown in the figure given in this figure. So this one, that A is the primary level, D is secondary level, C is tertiary level, and and B, where it is, we can see that two polypeptides are there. Is it two polypeptides in their tertiary level? So this one is fourth level or quaternary stage. So here we will see A is primary. First structure A, this one cannot be here. It is A writing fourth level. So this one cannot be. D also can't be. So our choice will be between A and C. Then B is giving secondary structure. No, it cannot. B is the quaternary structure. That is the fourth level. So our answer is C. That one is fourth. C here is the third degree of organization, and D is the second degree of organization. Like in case of our that human, very good example is a human, in the human blood, that hemoglobin hemoglobin is a protein, okay, that has the fourth degree of organization. And here what happened, we have hemoglobin is made of 4 polypeptide. Okay, 4 polypeptides are there in the hemoglobin, and these 4 polypeptides of course are in 2 different types. 4 polypeptides, but 2 different types of polypeptides. And 2 different types are, 2 are alpha type, 2 are alpha chain, alpha polypeptide, and 2 are beta chains are there. Beta polypeptides or beta chain. So altogether 4 are there. Okay. So this is the fourth level. Okay. Today I will stop now. I think it is a time excess. So I will close it. I will continue in the next class also this biomolecule. Okay. So I will
Thank
close
you.
it here. If you have any question, I think it better you ask in the next class. You, by the time you try to solve the question related to this, particularly proteins, for the next class.
Thank you,
Thank you,
sir.
sir.
Thank you very much.